What's going on, people? Georgio Savvides here, founder of the Hip Hop Orchestra, as well as the host of the Hip Hop Orchestra Presents podcast. And I got a really exciting announcement to finally make with everyone, is that we have our very first official sponsor for the podcast. Now, they are an amazing company called Eldon's Vault, who are extremely dedicated in bringing you the very best in officially licensed merchandise from pop culture. And when I mean the very best, I mean the very best. They provide things like keyrings, caps, t-shirts, to Funko Pops, to statues from all over, such as DC, Marvel, Disney, The Mandalorian, Harry Potter, music artists from all over the place, and they are ridiculously good. I've got a couple of things to show you on offer at the moment. This trust me, their stuff is amazing. So, t-shirts, like I mentioned, like the one I'm wearing right now. It's got Rocky right here, which I absolutely love. Or, like I said, also got Funko Pops as well, 3.75 inches and 10 inches. I've got a couple of 10 inches right here for you. I've got Post Malone right here, and I've also got Biggie right here, who I absolutely love. These are so sick. Eldon's Vault, trust me, they are amazing. Now, I'm going to leave the link below in the video right here, and also in the description below as well in all future videos from the podcast. But trust me, Eldon's Vault are amazing. Website is www.eldonsvault.co.uk. Get in touch with them now and start buying ASAP. But also, before I forget as well, I just want to say the biggest thank you to Eldon's Vault for sponsoring the podcast. It means the absolute world to me. Thank you. And trust me, get in touch with them right now. And I hope you enjoyed today's episode. And peace. What's going on, people? Welcome to a brand new episode of the Hip Hop Orchestra Presents. I'm joined by an amazing guest today, Postdoctoral Researcher at Stanford Institute for Stem Cell Biology and Regenerative Medicine, Sabra Djomehri. How are you? Okay?
Good, great. How are you?
Yeah, not too bad, thank you. How's your year been going so far with this madness?
It's been an incredible journey. Starting, moving back to California in early 2020, just a month before COVID hit. Starting at Stanford, that was, always been my dream. It's been, a long time coming, but we landed a really, amazing position in, breast cancer research. It was basically, something always wanted to do more like interdisciplinary work, at the junction of bioinformatics and cancer.
Fantastic.
Yeah,
How's that been going for you so far?
it's been really good. Our lab is one of the, pioneering, research in breast cancer. We do all different kinds of cancers, but we're kind of pioneering new like sequencing technologies. And yeah, so we then a new technology that we have going is targeted chromatin ligation and epigenomics.
Hmm.
And basically different RNA sequencing and focused on single cell technologies.
That's
Yeah.
fantastic, because I know that you did biomedical engineering at University of Michigan before this. Is
Yeah, I
that
did.
correct?
Yes.
Yeah. And how was that for you?
It was really good. At Michigan, they're kind of also really, one of the top research institutions for like also cancer,
Hmm.
cancer and, understanding like cancer cell phenotype dynamics and how, cancer cells, change states
Hmm.
basically, and how they're escaping the cancer or, mechanisms. And the stem cells are basically the ones the cells in the population in the tumor that are maintaining the cancer. So we have to find out what are their driver mutations and driver signaling pathways to basically create better therapies.
Hmm.
And yeah,
That's amazing. And that was a 4-year course, was that correct? 4 years you were there?
Yeah, for about 4 years. Yes.
4 years. And at the same time, did you also do become a doctor of philosophy as well at the same time
Yeah,
whilst
it was
there?
Doctor of Philosophy. Yeah,
So was it
in pathology.
yeah, pathology, sorry.
Yeah.
Was it a joint course or was it two separate,
Oh, it was just one.
or
It was
just one?
yeah. Mhm.
That must have been incredibly difficult to do that though, doing that such an intense course.
Yeah, exactly. I mean, the PhD programs are very supportive at Michigan, and they're really highly collaborative with the cancer, the Rogel Cancer Center,
Hmm.
and with like other departments. There, I found it to be extremely collaborative, and helping the students to really progress and make an impact. You know, versus like other institutions, which I've been at. Other institutions maybe they're not as supportive, but yeah.
I'm glad you enjoyed it though. Do you still keep in contact with anyone from there as well to help you with your
Yeah,
current work? Yeah.
I do. I mean, I keep in contact with my old mentor, Selena Claire.
Hmm.
She's phenomenal. She was known for discovering CCN6 protein, and EZH2. So they're, one of the first to understand, or to conduct these experiments on microarrays, which you can understand, RNA protein expression levels. And these will, have been really critical in discovering new like proteins and other, led to other therapies. So, that lab was doing a lot of uh, mouse experiments,
Hmm.
trials, and doing therapies. I also did, in addition, addition to the mouse models, we did organoid
Mm.
models. The organoids will in the future be pretty critical as an alternative to like animal testing.
Hmm.
And so those allow us to just develop, rapid testing on little self-assembling tissue structures
Hmm.
that are 3D that you can develop like in the lab, and you can do a lot of therapy testing, and just, it's very high throughput compared to, yeah, mouse models.
That's amazing. When you say
Yeah,
rapid testing, how rapid are we talking? We say several days or weeks or?
basically days and weeks,
Oh,
because
really?
yeah, they're very quick. You can get a lot of information, a lot of data from it very quickly. But you compare that to like mouse models where you have to develop them over like months.
Mm.
Yeah, so it takes a long time.
Is that because you work with organoids as well? Is that a lot easier to work
Mhm.
with than mouse trials? Do you feel?
Oh yeah, I mean it's necessary at the moment to have both to
Mm.
validate your therapies. But yeah, the it's just so tedious when in the lab when you work with, you're living with, yeah, working with a living thing. So it's like, you need to develop all these other protocols just to handle, if something doesn't work, and if you're really seeing the outcome that you want. But in the lab, when you have like a very simple array plate,
Hmm.
and you have like different wells, you know exactly how many wells there are, exactly how many cells you put into it, and then you're just tracking it with imaging or with different, you're collecting each tiny well, that information, and yeah, just running a screen basically
Mm.
on it. So
That's brilliant. That's fantastic. And when you do come across something that you're looking for exactly, man, what's that feeling like? It must be an incredible feeling.
oh yeah, it's pretty incredible. I have to admit, because when I started, I came from engineering
Hmm.
background and physics, and so I was like, oh, I don't know what I'm doing in biology, but I was very skeptical that anything's working because, when I started, it was just pipetting very small volumes of clear liquid that looked like, oh, what's in there, But then after like 48 hours, you could see the by fluorescence imaging.
Hmm.
You could see things actually changing in the structures of these organoids, and it was really exciting.
That's amazing. I know that
Yeah.
in the late 90s, when,
Mm.
'cause I obviously do a lot of work with stem cells, obviously. I know that, was it in 1998 where they first isolated human, what was it, human cells, was it, back in 98?
Yeah, and basically, once we immortalize the first like human cell line of the HeLa cells, after we discovered like the PCR and all these reverse transcriptase and all these things, it really like enabled us to develop all these different human cell lines,
Hmm.
and then we could just grow them in the lab indefinitely. Um, it's just that those cell lines are not as reproducible for the actual um, patient cells, because it's a lot harder to grow those in the lab because they're not indefinitely being produced. But now we're trying to go back and see, oh, what are the differences between those initial cell lines that we have generated to like actual patient information. So we have to then validate that again. So
Yes,
it's a big process. So
of course, can imagine. Because I
yeah,
did want to ask you actually how it works with different patients, because obviously everyone is different. I mean, what do you
yeah.
do when you say inject stem cells, for instance, into a different patient? Do you have to do several different in-depth tests for them to make sure that when you are injecting with the cells, they actually can take it properly?
Oh, of course. Yeah. So there's all these protocols to be able to make sure that it takes it. And then, when you inject cells, we first tried, in grafting those cells in mice, and it's usually, around 20% or 25% that it takes. And then once, so that's why you have to do a whole experiment, very tedious, with many, many mice, just so that a few could develop those like tumors, for example.
Mm.
And then we're only going to test on those. So it's, yeah.
Mm. You mentioned tumors there as well. I know that one of the early experiences on rats, I think rats and mice, where they're using undifferentiated cells. Is that correct? Where when you're injecting
Yeah.
those, it would cause, are they teratoma tumors? Is that correct?
Yeah, they're like actually, yeah, those undifferentiated cells are like, or the immature type
Mm.
cells are the stem cells. And so that's what we're trying to do in lab now. I'm trying to really focus on what are those phenotypes, or what are those states, because it turns out that there's not just one state that it could be and differentiated.
Mm-hmm.
It could potentially be multiple undifferentiated states, and then they can. It's like a shapeshifter, sort of like they
Mm-hmm.
can become uh, with different stimuli. They can become different things, and so we're trying to uncover what those states are, so that we can like develop therapies to target
Mm.
those. Yeah.
That is what I read up on, that these undifferentiated cells can become literally anything. You put them obviously with them work to it. But
Yeah.
when you, am I correct in saying that there's two main cells that you work with? You've got the adult cells and the embryonic stem cells as well. Are they the two main that
I
you
mean,
work with?
well, yeah, like we do those in the lab. Um, I'm not currently doing that, but yeah, the embryonic stem cells versus the like the other breast stem cells. So I'm mainly working with that, like breast cancer stem cells.
Mm.
And so exactly, it's like they can't quite become anything, but we're really honing in on what they can and can't be. But that's exactly what you touched on, is called like cellular plasticity.
Mm-hmm.
So that plasticity is like allowing it to become, have a tendency to become more things. So yeah,
Mm.
I'm pretty much working with primary cells, which are the patient derived cells, and running the screening on those.
Okay. How's that been going so far? It must be incredibly difficult to work on.
Yeah, so it's incredibly difficult. At the moment, because of COVID, actually,
Mm.
I'm focused on computational analyses. And so once you can have like large data sets, doing big data, you have to find those cell states by deconvolution
Mm.
through this, the data sets that you have, to be able to find those phenotypes. So I'm doing a lot of like bioinformatics, and like trying to apply, machine learning algorithms to these like data sets,
Mm-hmm.
and go from there, and trying to then at the end, like I've already developed a framework to look at uh, the genomics data, like whole exome sequencing. I don't know if you've
Wow.
heard about, yeah, whole exome sequencing is new. and then compare that to RNA sequencing, and then look at it on a single cell level um, to discriminate those like tumor subclones that are associated with the undifferentiated cells.
Mm. I
So,
would love to hear more about that framework, though. I didn't know that it was done at the moment. So, would you mind sharing a bit about that so
frame,
far? Yes, please. Is that
yeah,
okay?
yeah. So the framework is basically just a computational framework.
Mm.
So no one's, well, there's been a lot of effort now. It's very popular in the literature to go into ways to integrate these different data sets. For example, how do you combine actual intensity values from like an RNA to a protein level? And it's really, from the same cell, it's really hard. So the ways that we're trying to do it now is just to combine the two data sets
Mm-hmm.
into the single analysis, and standardize those results in a way that makes sense. So
Mm.
if you're looking at some mutation that you got from the same cell, and then you also had like RNA levels of it, then you can sort of put them in the same space
Mm.
analytically, and see how those patterns of RNA are up or down based on its mutational pattern.
Mm. Wow,
Yeah.
that is incredible. It really is. And I want to go back to what we kind of did earlier, where you mentioned sort of proteins in cells and so forth. From what I understand, I know that every cell has its own protein. And when you kind of isolate sort of the cells, I know that stem cells are obviously they're way more rare than, say, regular cells. So, like, say, regular cells got one every 100,000, is that correct? And
Yeah,
then
it's
bone
basically
marrow
like the
cells?
stem. Yeah, exactly. Like the
Yeah.
stem cells are like maybe account for 1% of the
Wow.
whole population, but you have stem cell population in every organ and every
Mm,
tissue.
of course.
So, and they could be in cancer, they could be recruited to like to a different location, a different organ. Like in breast cancer, it can be stem cells from the bone marrow can be recruited to the breast, or the brain, or the lung, and that's what enables the cancer metastasis.
Hmm.
When it gets really bad, then it can those stem cells can be also recruited different parts of the body, and then it's like spreads.
Mm.
So we're trying to understand that as well. But
That's definitely,
yeah, that part is so complicated.
of course. From what I understand as well, is that you can kind of isolate or find these specific cells by some kind of
Mm.
fluorescent light or so forth. Is that
Yeah,
correct?
yeah, we did let confocal microscopy.
Okay.
There's a whole heap of them, but fluorescence microscopy.
Mm.
So we do that by we can isolate it by staining, a staining procedure, but you have to then fix the cells. You have to isolate them first, and section them, and then
Mm.
once you section them, then you stain it for a particular protein, or whatever antibody, and then you can look at that particular one under the microscope. And it's really like high powered. It's extremely amazing, the type of level of detail that you can find with these imaging results.
Of course.
Um,
And I can
yeah,
know that obviously with stem cells, they kind of hide between other cells. Is that correct? Where they kind of
they,
hide or kind of
yeah, I
camouflage
guess they
themselves in
can.
between. Yeah.
They kind of
So,
camouflage themselves, yeah. But the good news is that if you have the right marker, or the
mm,
protein marker, you can like uncover it in a way that it's not hiding anymore, and you can see that distinguished from all the other cancer cells, and you can actually see the spatial location of like where it would be hiding with respect to the other cells. So
mm.
it's pretty cool that we can.
No, no, it
Yeah.
really is. Because when I was looking into it again over the last couple of days to kind of refresh my memory on a couple of bits, obviously
Mhm.
I'm nowhere near your level, but when I read into it, it really fascinates me how in depth it actually is. And I
Mhm.
know that obviously they're much rarer than, say, regular cells or bone marrow cells, but I know that certain problems within the body are due to lack of cells. Say, for instance, lack of heart cells cause heart trouble, to lack of dopamine causes tremors, to Alzheimer's and so forth. Why why does the body not create? I know it's a bit of a stupid question, but why does the body not or can't produce more stem cells? Is it because we simply don't create enough as we get older, or do we like lose them as we get older? How does it all work?
Yeah, so I mean, but from embryogenesis, basically, it's just every cell in the body is programmed to have a certain amount of stem cells. So that's all set in stone basically from birth or from in the womb. But then like as you get older, it's things are happening that are just as a result of the signaling pathways, for example, that are so what happens is that your cells, when they proliferate more, they actually the cell is bigger.
Mm.
So as you get older, like I guess you could maybe talk to an aging researcher, but it's also connected to some cancer pathways. But the RNA content is like increased.
Mm-hmm.
But then basically, when you get older, it's like its ability to regulate those pathways, is less effective. And then it kind of would complicate certain things, and then it'll, as a secondary signaling result, it'll drive other factors that could lead to diseases.
Mm.
So that's you know. Um,
Okay. One thing I was a bit confused about, obviously a lot of it did obviously confuse me, but the one thing I wanted to ask you actually directly was, I know that with adult cells, you can take them both from both adults and infants, yet they're still called adult cells. Why is that? Is that because the cells themselves are fully
okay,
formed, or
the
is it
adult
a different
cells,
reason? Yeah,
they're,
the adult
I
cells, yeah.
mean, they're kind of like we would consider that like the mature
Mm.
cells or the differentiated form. And so, when we look at it, like we'll analyze the undifferentiated versus the differentiated form. So
Mm-hmm.
it's, there's only, so those cells really can't become anything else,
Okay.
and then they have a set, time limit and then for cell death. So yeah, did you have a question about how that is, or?
Yeah, I wanted to ask you because you mentioned the mature cells, which I did want to ask you as well. Do you know how many variations of mature cells there are available and what they kind of do as well?
Yeah, it's pretty specific to the tissue or the organ that's sitting in. So like, in the breast, it's like the lobules and the duct.
Hmm.
So those are those are the two main ones,
Okay.
and yeah. So it's pretty well known. It's just based on the anatomy. Those adult cells are easy to track. That we know their markers very clearly. So then it'll help us like backtrack for
Hmm.
the stem cells. Yeah.
Okay. And does every cell have the three layers? So the mesoderm, the exoderm, and the endoderm. Do they all have three layers, or
I
do every
mean,
cell?
not
Sorry,
yeah,
go on.
like every cell has derives from one of those three
Okay.
layers. Yeah.
Okay. So I wanted to ask you as well if you're able to say use each part of the layer for different things, if possible, or do you need to use a whole cell to create what you need?
A
Say
whole cell.
cartilage, or say like cartilage,
Oh,
or anything like. Are you able to like say
yeah.
like dissect into three
Mhm.
layers if there are three layers available, obviously, or do you need to say the whole cell to create exactly what you need for this specific
I mean,
reason?
for the germ layers, you would just need, you couldn't get all the three,
Mm.
but basically, from doing the experiments, you would definitely need the whole cell. And then you need to like uh, isolate those cells and know exactly what you have. And so that you could analyze what protein is expected to give you some profile that
Mm.
for the therapy that you're after. So
Okay. And am I also correct in saying that when you do, say, inject the stem cells into, say you got torn ligaments in your shoulder, for instance, do you need to directly inject it into the affected area, or can you just? I know it's a bit of a silly question again, but do you need to inject directly into the affected area, or can you inject it, say, anywhere? Like I know with diabetics, you can say inject in the shoulder or the legs or
yeah,
anywhere else.
sometimes they do both because when you're what you're saying, like if you have like a localized like
Mm.
injury or some type of localized thing, then you should inject it to that site for sure.
Mm.
But then there's other things that are maybe diseases like with the vaccine, for example, that you can inject it, and then it doesn't have to be specific to that site,
Mm.
because the effect is going to be systemic. So then, yeah, so it's just the difference between systemic versus a localized therapy, and
Okay.
it's just, yeah, specific for that condition.
Mm.
So
I saw a video recently where they were taking fat from either the buttocks or the flanks area of the body to harvest with the cells. Why do they do that actually? Why do they mix the cells with the fat? Does that help kind of reproduce more cells or something like that?
yeah,
What's the reason for that?
I mean, it would be pretty specific, but for example, breast cancer, they inject it into the like mammary fat pad.
Mm.
That's just the gold standard way to do it. But because the fat cells are a cell compartment that has a huge impact on the maintenance of what's going on in that whole tissue.
Mm.
So what like the dynamics of the whole like um, signaling and all the events. And so, yeah, I guess, they found probably decades ago that injecting it into the mammary fat pads or other, fat cells are like more efficient to have that desired effect for it to spread.
Mm.
So, yeah.
Okay. And when you do take the cells from, say, the liver, for instance, or wherever it may be, do you have do you store them in different ways, or do you have to store them in the same sort of way? So, for instance, do you need to freeze them immediately, or do you need to keep them in at, say, room temperature to help reproduce, for instance? How do you keep the cells when you take them?
Yeah, so it's pretty much a very similar way when you take different types of cells. We freeze it, we put it in a freezing media, DMSO.
Hmm.
But you don't have to if you want to. That's if you want to preserve it. But if you want to just grow them directly, you don't have to freeze it. You can just grow it
Oh,
on a plate and then keep them growing as for indefinitely. And usually if you want to do all these experiments, then it's best to just but you need a specific like solution or liquid that it has a specific set of like nutrients for those cells so that
okay.
those cells can grow up,
Okay.
um, yeah.
Okay. And when you, and I'm really, I want to ask a million questions, but I know you're really busy, so I
Yeah.
won't keep you for too long. But I know with injections, say, depending
Mhm.
on what the patient actually needs, say the regrowth of, say, spinal column or vertebrae to a torn muscle, for instance,
Mhm.
is it, say, a one-off injection? Another bit of a silly question again, but is it just a one-off, or do you need, say, several injections to really get the desired result?
Well, I think it depends, but I definitely know that there should be a regimen of like a series of them for the yeah. That's what I know about, but like in our mice studies, we have like several. We have
Mm.
like a whole protocol that day one it's going to be a certain injection of the cells, and then we wait, and then we analyze like how big like a tumor has grown, for example, and then we know exactly the time frame that we want to start injecting other dosages of like a therapy after several days apart, and then we test the effects that way. But yeah,
Oh,
usually it's a series of
okay.
injections. Yeah.
And through that series, is it, say, once every couple of weeks or once a month? How do you kind of pan out your sessions?
Yeah,
Or
I
does
think
that
it's
it depends
usually
on the patient?
like
Yeah.
depends on the patient, but like
Mm.
probably weekly. But yeah, like that's definitely more like clinical oriented. Um, yeah, I would speak to a clinician about the exact
Oh,
regimen, but it's
okay.
it's so different. I know it's very different when you actually look at the cells, from the patient versus the patient itself. It's like it has a profound, the effect, like different. So everything is, that's one thing that's very challenging with biology experiments to get it right, because it's like every system is so particular and you need a protocol for each one. So
Of
we're still
course.
working on that. So
Okay. I want to ask as well about what the patient needs to do once the injection has been taken. Do they rest or can do the daily routine? But I'll ask the clinician for that if you want me
yeah,
to.
after
Yeah.
going,
No,
yeah,
I will do, don't worry.
yeah.
But I wanted to ask, do you tend to work with the ISSCR by chance, or the International Society of Stem Cell Research? to help with your?
Oh, no, I do not.
Okay.
Yeah. I mean, we work with, our cancer center that's in our institute. So it's like the Stanford Cancer Institute. But yeah, we do have other, laboratories like the Jackson Laboratory that we get everyone gets, their mouse models from there, for example. Or you can custom make your own in the lab. But there's a lot going on in Stanford with like, custom developing platforms for like sequencing technologies. But yeah, it's collaborating and a lot of grant writing with like NIH, NSF and stuff. But probably in the UK it's like a little different. I know it's pretty different.
Yeah, I can imagine. Talk about grants actually. How difficult? I know recently, I think, was it in California where they gave I think $4.5 billion grant or funding for stem cell research. Is that correct?
I
Or around
forgot
that?
the exact name of that, but yeah, it's yeah, exactly. It just passed, right? Um,
Yeah.
yeah, so 4 billion, and that basically was directly impacting our research institution. So we were extremely happy about that. Yeah.
That is amazing. But with these cells that you directly work with cancer as well, I know that there's kind of different potency levels or sort of developmental capacity, so multipotent, pluripotent, and totipotent.
Oh yeah, exactly. You hit
Yeah,
it.
no, was that
Yeah,
correct? Yes.
exactly.
Okay. Oh, brilliant. Yes.
Yeah.
When you do the stems or the cells you work with
Mhm.
have all three levels, or do they have a one specific kind of potency level, really? And
Yeah, so
what?
we're actually trying to like understand that now because
Mm,
those terminology that you just mentioned was specific to the basic, three like the germ layers and like what we know biologically to be true based on those germ layers. However, in cancer, it's like, for example, in other diseases, it's really difficult to know how those states are they the same in cancer? I think we think that it has multiple within each of those that you just mentioned. So the multipotent versus the totipotent or the unipotent. So we think now that in cancer, it's like we discover a few different states that are distinct
hmm
within each of those categories. So we're trying to like see, oh, what are the differences? Which ones are more potent than others? But
mm.
we really don't know at this point. But it's really exciting to be in that kind of lab that we're uncovering that. Um,
Okay, because you mentioned unipotency as well, which we're going to get into.
mhm.
Does unipotency and also oligopotency, are they kind of subdivisions of these three, or are they kind of just their
Yeah,
own separate
no, they're all
entities?
distinct. Yeah, they're separate entities, basically.
Oh, fair enough. Okay. Um, I want I'll ask a couple more questions. I've got loads more, but I know
Yeah,
you're busy, so I'll be quick then. So I
yeah.
know that with embryonic stem cells or ES cells, from what I understand, they're a very controversial thing to work on. Is that? Are you
It's
able
kind
to
of, I think it's
explain?
less controversial now, but yeah, I mean, yeah, because sometimes like, in different countries also like that you derive those cells from different sources, so maybe that's why it's controversial. But it's all that's why we're able to like now study like adult stem cells as well. Like we don't need to um, derive our stem cell populations from embryonic anymore. Some
Mm,
people do. We have created uh, cell lines to be able to get those embryonic stem cells. So it's
mm,
just like mass produced. So we don't need to get it from any other sources. We can just, buy it directly from the vendor. So it's nice and
okay.
easy. Yeah. But
Mm,
yeah, so but the adult stem cells, which are in every organ, we can actually extract those directly. So it makes it a lot easier to understand.
yeah, 'cause I did
It's less
see that.
controversial.
Obviously, I know that from what I understand is
Mhm.
to the kind of to the inner cell mass from the blastocyst, and obviously that destroys the blastocyst, which is what I understand was the
Exactly.
contribution of why it was. But from obviously over the last few
Mhm.
years, has that have you been able to kind of create something new without being able to destroy the blastocyst?
Oh yeah, because that was
Cyst?
initially what they use, but then the iPS cells, which you can, so we don't need to derive it, our stem cells from that and like anymore. That's just one way to do it. So we can get it from other tissues, directly from patient tissues that are patients like that are current in the hospital, for example, from biopsy samples. You can also extract it there. You can also induce, like based on that plasticity that I mentioned,
Mm-hmm.
you can get more adult stem cell populations, and then you can reverse them to be undifferentiated, to be more
Okay.
totipotent. So that's another way to, that was like, one of the breakthroughs as well. So
Okay.
yeah,
I know, is totipotent also water-based, from what I understand as well?
water-based.
Someone's told me that totipotent is
Mhm.
a water-based sort of level, but if that's the wrong case, I'll just leave that
Maybe
there.
that's it. Yeah,
Yeah,
yeah.
I'll leave that. um, but I know that obviously with blastocyst as well, it's obviously it's pre-implementation embryo that develops 5 days after fertilization. Are you able to use it after the 5 day period, or is that 5 day, sort of 5 days
Yeah,
after?
you can use it at any period,
Oh, you
basically.
can.
Yeah.
Okay.
Mhm.
And what something I was slightly confused about was in the description of in vitro and in vivo as
Oh,
well. Yeah.
yeah. For example, the mouse studies that we do are called would be in vivo because they're living, um, like a living creature that you're utilizing for
Mhm.
experiment, versus in vitro, which doesn't use any type of like animal tissue,
Hmm.
and nothing living. It's just in the lab. Anything that you grow, like cells that you grow in a lab, um,
Okay.
that would be like in vitro.
Okay. 'Cause
Like
I
the
wasn't.
organoid would be like an in vitro model. So that's another.
Okay.
Yeah.
I was trying to read up on it, but I kind of got a bit
Mhm.
lost in that as well, unfortunately. But
Exactly. Yeah.
do,
That's
do you,
okay.
but do you work with spores by chance as well in the work that you do?
Spores? No, I don't currently.
Okay. What are somatic cells within this? I know that I've heard about somatic cells. What are
Yeah,
they
they're just the
exactly?
adult cells, like, um, you know, yeah, anything that's not for reproducing cells, basically. Yeah, they're just all the cells, like adult cells.
Oh, okay.
So
I found. And do you work with nuclear transfer as well by chance?
no, I don't, but yeah, I know a little bit about it, but
Okay, because I
yeah.
know that that's part of the ESL, so I was
Oh, okay.
going to see if you kind of work with that within your work.
Mhm.
But I know it's a random question, but do you ever talk about Dolly the sheep by chance as
I
well?
mean, I mean, not really, just maybe in the, biology 101 course,
Yeah,
but
fair
yeah.
enough. But before you go, I want to ask, what do you feel is the future of stem cells? Do you feel, especially in your
Yeah,
cancer as well?
I hope the future is developing novel therapies that would target stem cells, and being, in the next decade that we can get something that's pretty, strong evidence that it could work in clinical trials.
Hmm.
Um, that would be my, yeah, number one.
Fantastic.
Yeah.
I never asked you actually, because normally I like to start off my podcast by asking how you got into your work. So, what really drew you into wanting to work with sort of cancer, stem cells, and so forth? What really drew you into it?
Yeah, I was, inspired early on because my mom actually passed of breast cancer
Oh, I'm
in
sorry to hear.
2004. I got really inspired after that. I was like a college student at the time, and
Hmm.
I was more on the engineering side. I didn't know what I wanted to do, and then I had another family member, sibling who had like a heart surgery, and then I started to understand more about these novel devices, like biomedical devices that are like revolutionizing some treatments. So that's how I got into the engineering side of it, and then after that, really honing in on the cancer. So yeah, it's taking off, and we're excited.
Oh, brilliant. But Sab, I just want to say thank you so much for taking the time to speak with me today. Honestly, I could have spoken for so much longer today, but I know how really busy you
No,
are.
it's
But
really
thank you.
interesting.
Yeah,
Yeah,
but thank
thanks
you
so
so
much.
much for
Yeah,
today, honestly, and I hope we can catch up soon, man. Best luck with everything in the future as well.
Thank you so much.
Thank
Thanks for
you.
having me.
My
Yeah.
pleasure. And where can people find you, by the way, online, just in case?
Yeah, that Stanford Profiles is my website. That's the best, place to reach me. You can contact me, and for
Brilliant.
any questions you have. Yeah.
Lovely. Oh, two quick things actually.
Mhm.
I see that you've written 13 journals as well within your profile. How was that for you to write 13 journals?
Oh, I know. It's been really amazing. It's just taking off more, with the, the work that we've been doing and the recognition for, all the citations. So yeah, it's been really exciting now to be able to publish in different areas, like quantum, or not, I'm sorry, the quantitative, that's what I'm getting into next, actually. But basically, the computational aspect and comparing it with all the in vivo work
Mm,
is something that I'm really excited with.
that's amazing.
Work, so
That's
yeah.
amazing. And the last question for now, I did obviously other questions, but I don't want to keep you any longer,
Yeah.
is do you know anything about osteoporosis as well, with stem cells as well?
Uh, I know a little bit about it. I used to do, some work in the micro CT, like imaging facility
Yeah.
at UCSF, but I don't know too much about the osteos, side of that with the stem cells. But
Okay.
yeah.
Oh, because my mother suffered really badly from it, so I was going to see
Oh
if you
no.
not
Yeah.
see if you maybe knew of anything or details if you could
Okay.
help or not. But
Yeah.
no worries, that's fine.
Yeah.
But thank you so much again for today, Sabra, and I hope we catch up soon.
All right. Hope to
Hey,
catch up soon.
thank you. Take
Thanks.
care. Bye bye.
Take care. Bye.